Hello and welcome to BioDigest. If you can't fly, run. If you can't run, walk. If you can't walk, crawl. But always keep moving towards your goal. With this beautiful quote, I welcome my students to the biology class. In the previous video, I was discussing about osmosis. We will continue with the same topic. So moving to osmosis again. What is osmosis? It is the process by which water molecules will be moving from the region of its higher concentration to the region of its lower concentration through a semi-permeable membrane. In the previous video, I explained this process by using a potato osmoscope. Now one more experiment we will see with regards to osmosis process. For that, I am going to take one thistle funnel like this. Okay, just drawing the thistle funnel. This is what is known as a thistle funnel. This is the mouth of the thistle funnel. Now the mouth of the thistle funnel is actually covered by using a semi-permeable membrane. We can use cellophane paper or an egg membrane. You all might have seen the egg membrane. When you boil the egg and when you peel off the shell of the egg, there is a transparent membrane that you can see. That is what is known as egg membrane, which is actually acting like a semi-permeable membrane. So what is a semi-permeable membrane? A semi-permeable membrane is a membrane which allows only certain molecules to pass through them. So I told you the mouth is actually covered by using an egg membrane. Okay, so this is the egg membrane. Now after covering the mouth of the thistle funnel by using an egg membrane, what we have to do is we are filling this thistle funnel with concentrated sugar solution to a certain level. Okay, like this. This is concentrated sugar solution. A concentrated sugar solution can be made by taking 100 ml of water and adding 3 teaspoons of sugar to it. So we are filling that sugar solution into this thistle funnel up to a certain limit. Then the level of the sugar solution is actually marked by using a marker pen like this. So this is the initial position of the sugar solution. Initial position means the beginning position of the sugar solution. Now what you have to do? We are actually taking a stand like this. Okay, this is a stand. Now onto the stand we are placing a beaker that is containing pure water. So water is taken inside the beaker. I am shading the water like this. So this is water. Now what you have to do? Bring this thistle funnel containing the concentrated sugar solution and place this in this beaker containing water like this. So here I am little bit rubbing that shaded water for you to identify that. Otherwise the thistle funnel will not be visible. Okay, so this is the situation. See. Now what is there inside this? There is sugar solution like this up to a certain limit. Now there will be, of course, there will be a clamp that will be keeping this thistle funnel in position. Now keep this experimental setup like this for 1 hour. What will be the observation after 1 hour? Again, this is a board exam question. After 1 hour, you will be observing that there will be definitely an increase in the level of sugar solution. Okay, I am shading that much portion. So the dotted one, this is the initial position. Okay, the beginning position of the sugar solution. Now after 1 hour, you can see that the level has increased. So this is the final position. So why there is an increase in the level of sugar solution within this thistle funnel? What could be the reason? Of course, it is because of osmosis. Why? Because I told you there is pure water that is present within this beaker. And there is concentrated sugar solution that is present within the thistle funnel. So always water is having a tendency to move from the region of its higher concentration to the region of its lower concentration through a semi-permeable membrane. So water that is present outside will be moving into this thistle funnel through the egg membrane that is acting like a semi-permeable membrane. As a result of which the level of the sugar solution in the thistle funnel will be increasing. So here actually the inward movement of water is taking place. This is what is known as endosmosis. Okay, now there can be a control experimental setup. What is a control experimental setup? An experimental setup wherein the variable that is being tested will be missing is what is known as a control experimental setup. So here we can use the same thing, but there will be one change for the control experimental setup. Inside the thistle funnel, instead of filling this thistle funnel with the sugar solution, we can fill the thistle funnel with water. Okay, so this will be the control experimental setup. Water will be filled inside the thistle funnel. And again the same experimental setup, again I am showing the same. So we will rub this thistle funnel containing water now. Again I am going to show this by using the second control experimental setup. Okay, so here what we have to do, there will be a stand like this. There is a beaker containing water. Then there is the thistle funnel that is filled with water to a certain extent like this. Of course, the mouth is covered by using the egg membrane. And instead of filling it with sugar solution, we have filled this with water. So water is present inside and outside. Okay. Outside this also contains water. Inside the thistle funnel also water is present. So we are taking water inside and outside. So this is the experimental setup. This is what is known as a control experimental setup. The variable that is being tested will be missing. Okay, the variable being tested is missing is what is known as a control experimental setup. So here we can see two experimental setups now. One is the thistle funnel is filled with sugar solution. In another case, we are filling the thistle funnel with water. Both we are keeping it for 1 hour. Let this be marked as A and this be marked as B. This is experimental setup A and this is experimental setup B. What will be your observation? In experimental setup A, there will be an increase in the level of sugar solution after 1 hour. But in experimental setup B, the level of the water inside the thistle funnel will remain unchanged. What is the reason? It is because inside the thistle funnel and outside the thistle funnel it is containing water. So water will not be moving from inside to outside and outside to inside. So that the level of water inside the thistle funnel will remain unchanged. That means no osmosis will be taking place in this particular case. So this is what is known as a controlled experimental setup. Moving to another definition. Osmotic pressure. What is osmotic pressure? We will be writing it as OP. So osmotic pressure of a solution is the measure of its tendency to take in water by osmosis. Okay, the solution is having a tendency to take in water by osmosis. That tendency of a solution is what is known as osmotic pressure of a solution. Now moving to another definition, that is tonicity. For better understanding of this particular topic, we will compare tonicity and osmosis together. Okay, so we are having two setups like this. In osmosis, actually the diffusion of water across a semi-permeable membrane is taking place. Okay, so this is one solution, this is another solution. The diffusion of water molecules across the semi-permeable membrane, that is what is known as osmosis. The water always moves from the region of its higher concentration to the region of its lower concentration through this semi-permeable membrane. It is what is known as osmosis. Whereas in tonicity, we are actually considering the relative solute concentration of two environments separated by a semi-permeable membrane. So we are considering the solute concentrations, okay, of two environments like this, separated by a semi-permeable membrane. See, so this is actually the solute. These are the two environments. So we are comparing the solute concentration of two different environments separated by a semi-permeable membrane. That is what is known as tonicity. Okay, so I hope the difference between tonicity and osmosis is clear to all the students. In osmosis, it is the diffusion of water molecules across the semi-permeable membrane that is considered. Whereas in tonicity, the relative concentration of solutes in two different environmental conditions separated by a semi-permeable membrane is taken into consideration. So based on this, there are three types of solutions. Which are they? Isotonic, hypotonic and hypertonic solution. Coming to the first type, that is hypertonic solution. Hypertonic solution means the solution will be highly concentrated. That means it will be having higher amount of solute. So we are having a highly concentrated solution here, which is prepared by using sugar or salt. In this, I am going to place a plant cell. So simply drawing the diagram of a plant cell for you to understand it. I am rubbing this. So this is a plant cell. Cell wall, cell membrane, nucleus, then organelles. Okay. So this is a plant cell that is placed in hypertonic solution, which is a highly concentrated solution. In this case, what will happen? So here again, inside the solute concentration will be different. Outside the cell, the solute concentration is different. So usually, where the solute concentration is less, the water from that area will be moving to the area where there is more solute concentration. So here, I told you that the cell is placed in a highly concentrated solution. That means the solute amount will be higher outside than the inner condition of the cell. In that case, what will happen? Water will be moving from within the cell to outside. That means X-osmosis will be taking place when a cell is placed in hypertonic solution. So as a result, what will be happening to the cell? The cell will be shrinking. Shrink means it will reduce its size. So that is exosmosis. Exosmosis will be causing the shrinkage of the cell. So that is about hypertonic solution. Now moving to hypertonic solution. What is a hypertonic solution? Hypertonic solution means the solute amount will be very less. Okay? So in hypertonic solution, what will be happening? So here this is a hypertonic solution which is having very less amount of solute concentration. That is hypertonic solution. So if you are placing a plant cell like this in a hypertonic solution, what will happen? Here in hypertonic solution, endosmosis will be taking place. That means water molecules will be moving from outside to inside of the cell because there is more solute present inside than outside. So in that case, water molecules will be moving from outside to inside of the cell. As a result, what will be happening to this plant cell? The plant cells will be getting bulged, or maximum it will become turgid. Turgid means this enlarged condition. The cells will become enlarged due to the entry of water. That is actually happening in hypotonic solution. Then what is iso? Iso means same. That means in this beaker we are having a solution and a plant cell is placed in that. So what will be happening to this? Outside and inside the solute concentration will be the same. That is what is known as isotonic. Iso meaning equal or same. So here whatever is the solute concentration outside the cell, the same will be the solute concentration within the cell also. So in that case, what will be happening? No net movement of water molecules will take place. That means no osmosis will happen. So if no osmosis is taking place, what will be happening to the size of the cell? The size of the cell remains unchanged. Okay, it will be the same itself. So the size of the cell remains unchanged. That is isotonic. So this is hypertonic solution where we are placing the cell in a highly concentrated solution. So outside is having more solute than inner side of the cell. So in that case, water will be moving from within the cell to outside. Exosmosis will be taking place, which will lead to the shrinkage of the cell. This is hypotonic solution. Hypotonic solution means a solution will be having more solute concentrate within the cell than outside. So outside this solution is having very less solute concentrate. So in that case, what will happen? Water will be moving from outside into the cell, as a result of which the cell will be getting enlarged. That means endosmosis is taking place. Now coming to the last solution, that is isotonic solution. Iso meaning equal or same. So whatever is the solute concentrate within the cell, the same will be the solute concentrate outside the cell also. So in that case, no osmosis will take place. So the size of the cell will remain unchanged. So what is the difference between osmosis and tonicity? Osmosis actually refers to the diffusion of water molecules across a semi-permeable membrane. Then what about tonicity? Tonicity is actually the relative solute concentration of two different environments separated by a semi-permeable membrane. So based on that, we are having three types of solutions: isotonic, hypotonic and hypertonic solution.
Let us recall. What is osmotic pressure? Osmotic pressure of a solution is a measure of its tendency to take in water by osmosis. Tonicity, the relative concentration of solutions that determine the direction and extent of diffusion, is called tonicity. Based on it, the solutions can be of three types: isotonic, hypotonic and hypertonic solutions. Please do subscribe Bio Digest, like and share. Thank you.